So our next speaker is Dr. Larry Zweifel from University of Washington to speak about molecular and circuit mechanisms regulating GABAergic control of the ventral midbrain. Welcome.
Okay, thank you. Thank you to Sherry for the invitation, as well as the organizing committee. So I'm actually not going to be talking about imaging in live tissue. Instead, I'm going to be talking about images that are obtained from fixed dead tissue. But I will talk about some tools that we're working on that will provide means to image neurons that make specific projections. So what we're interested in is essentially what controls the ventral midbrain, and in particular, what controls the dopamine system of the ventral midbrain. And you'll hear more about this in Li Chen's talk probably later in the day. So we know that there's tremendous diversity in the connectivity of the ventral midbrain, and in specifically projections onto dopamine neurons. So we now have the ability to map inputs onto specific cell types using Cre-dependent conditional rabies virus tracing techniques. And Li Chen's lab has also beautifully shown that you can map inputs onto specific cell types that make specific connections to downstream targets. So in addition to these different connections, we also have different neurotransmitter-specific control over brain circuits. And in the ventral midbrain, we have specific neurotransmitter systems that play an important role in regulating the activity patterns of the cell. In particular, nicotinic acetylcholine receptors, ionotropic GABA receptors, and NMDA receptors, as well as a suite of ion channels, really control the activity patterns of the midbrain dopamine system. So taking these two components together, which is the diversity of the inputs to the dopamine system, as well as the diversity in the neurotransmitter control of the dopamine system, what we're interested in is what is the neurotransmitter or molecular identity of the different inputs onto the dopamine neurons. So to essentially address this question of, okay, we know different dopamine neurons project to different areas. We know that there are different areas that project onto these. What is the identity of these cells within a given area that is providing the neurotransmitter control to those specific dopamine cells? So to achieve this, we've developed a retrograde transducing Cre-dependent canine adenoviral vector system. So many of you might be familiar with the canine adenoviral vectors. It's a monosynaptic retrograde transducing viral vector, which allows you to selectively label inputs to a given area of the brain. So unlike the conditional rabies virus approaches where we're selectively isolating inputs onto a cell of a given molecular identity, what we want to know is the molecular identity of the cell that's synapsing to that region. So what we've created is a monosynaptic canine adenoviral vector system. We, myself and Richard Palmer, collaborated to generate a, I keep pushing the wrong button, a recombinant viral genome that contains a small number of multiple cloning sites, which allows us to rapidly generate different recombinant canine adenoviral vectors. So essentially, we created a two-step cloning process in which you have your recombinant canine adenoviral genome and a shuttle vector that contains a Cre-dependent gene of interest. Using a small number of restriction sites, and you'll have to do mutagenesis if your transgene of interest has these sites, you can clone it directly into the viral genome. So we no longer have to do recombinering into the viral genome, which is labor intensive. We can do direct cloning and then screen for the presence of ampicillin resistance and the loss of chloramphenicol resistance. So using this strategy in combination with the technique developed by Eric Kramer, which allows us to do transcomplementation in specific dog kidney cells that contain a critical gene that's required for the replication of the virus. We can transfect circular viral genome, induce its linearization using tamoxifen-inducible restriction enzyme, which then allows for the rapid amplification of the virus, and then we can purify it and then do molecular tracing studies. So our sort of proof of principle for this technique was to generate a viral vector which would allow us to do sort of a bird's eye view of inputs to the ventral tegmental area based on the different neurotransmitters that they use. So the first thing that we tested was serotonin neurons. So all of the inputs into the ventral tegmental area, the serotonergic system is the most dense, robust projection onto the dopamine neurons. So what we did was we injected the recombinant canine adenoviral vector containing a Cre-dependent ZS green into the ventral tegmental area of an EPAT1 Cre mouse. So EPAT1 is a specific marker for serotonergic neurons. It then allowed us to isolate specifically the serotonin projections onto the dopamine cells. And we then confirmed that these serotonin neurons, in fact, project there by labeling those cells with the synaptophysin GFP to label the terminals in the ventral tegmental area, and confirm that there is, in fact, a robust direct connection from the dorsal raphe serotonin neurons to the ventral tegmental area. Okay, so we confirmed that there are serotonergic projections to the ventral tegmental area. The next question is, what are the other projections to the VTA? What's their neurotransmitter identity? So the first one we did was the vesicular GABA transporter. So these are inhibitory neurons, and we expected to find a lot of local connections in the ventral tegmental area with a few long loop inhibitory feedback projections from the striatum. What we were surprised to find is that almost every area that had been previously identified using the rabies strategy as projecting to the ventral tegmental area is actually an inhibitory GABAergic projection. So nearly every area that projects to the ventral tegmental area is actually an inhibitory projection. And this is part of a larger scale study that we've been doing looking at the connectivity of the dopamine system in male and female animals. And rather not remarkably, the connectivity of the male and female ventral tegmental area is pretty much 95% concordant. So within statistical area, they have essentially the same density of projections from all of the same areas. So there is no real large sexual dimorphism in connectivity of the ventral tegmental area. So then the next thing we looked at was, well, if there's lots of inhibitory connections, are there an equal number of excitatory connections? And what we were surprised to find is that while there are numerous excitatory connections to the ventral tegmental area, and these are VGLUT2 positive cells. So previous anatomical ultrastructural data has demonstrated that the vast majority of excitatory input to the ventral tegmental area is subcortical VGLUT2 containing vesicles. And what we find whenever we label projections in a VGLUT2 Cre animal is that there are quite a few areas that are glutamatergic, and many of which we have already known about, including the lateral dorsal tegmentum, the dorsal raphe, the pedunculopontine tegmental nucleus. But what was striking is the relatively small number of these projections. So when we look at sort of the overall projection density map, and if we look at GABA and glutamate and serotonin, and we've also done cholinergic input to the ventral tegmental area, far and away, by nearly an order of magnitude, the largest number of inputs to the ventral tegmental area are in fact GABAergic projections from distal areas, not local GABAergic connections, although there are quite a few local GABAergic connections. So this was rather intriguing to us. So then we went on to say, okay, well, if there seems to be a large array of GABAergic input and some glutamatergic input, serotonergic, acetylcholinergic, is there any specificity in the types of inputs onto the dopamine system? So to begin to address this question, what we did was we isolated the polyribosome specifically from dopamine neurons using Stan McKnight's ribotag strategy, in which there is an affinity-tagged ribosomal protein. We can do immunoprecipitation of polyribosomes, isolate the actively translated mRNA, and look for neurotransmitter-specific genes and ion-specific genes that might be selectively expressed within the dopamine cells. And when we did this, what we found is there are a large number of neurotransmitter-specific genes that are expressed in the ventral tegmental area dopamine neurons, a large number of ion channels that are expressed in the ventral tegmental area dopamine neurons. But the greatest selectivity, which is genes that have the most enriched expression within dopamine neurons, are actually arising from the smallest input to the dopamine cell. So the cholinergic input was far and away the smallest input on to the ventral tegmental area, but it has the greatest specificity. So the largest number of ion channel receptor subunits that are expressed highly selectively in the ventral tegmental area. The second group of most highly enriched genes for neurotransmitters in the ventral tegmental area dopamine neurons are in fact neuropeptides. So many of the neuropeptide receptors for neurotensin, tachykinin, ghrelin, as well as dynorphin are also extremely highly enriched in the ventral tegmental area dopamine neurons. So although there appears to be a large array of GABAergic input, there appears that most of the specificity is likely to arrive from different peptidergic inputs from those GABAergic projections. Okay. So what do we have on the horizon for these different canine adenoviral vectors? So we've generated several. Those that are in the pipeline for behavioral characterization right now are the CAV2 Flex CHR2 YFP, which is shown here. So these are dopaminergic projections from the ventral tegmental area to the nucleus accumbens, so we can express channel rhodopsin in specific cells with molecular identity. We have a CAV2 Flex GCAMP6, which is also ready for in vivo testing, as well as HM4. These others are currently in the pipeline and undergoing viral production. So within the next year, we hope to have, including the ones that are already made, about 8 to 10 Cre-dependent canine adenoviral vectors that will be available for use. Okay, so that's our foray into looking at the different connectivity and the development of tools to look at connectivity of the ventral tegmental area. One of the questions that we're really interested in is what actually regulates the excitability of dopamine neurons and what actually regulates this neurotransmitter-specific connectivity of the ventral tegmental area. And these two projects sort of started independently and have now begun to coalesce around a specific project, which I'll tell you about now. So a few years ago, a postdoctoral fellow in my lab was interested in genes that may control synaptic connectivity in the adult ventral tegmental area. So what he did was he used the Allen Brain Atlas to look for genes that have unique expression patterns in the brain, particularly genes that appear to have some enriched expression in the ventral tegmental area. And what he identified are a large number of developmental axon guidance genes, of which here is a subset of axonal guidance genes related to netrin DCC and slit robo signaling, which is known to play an important role in the wiring of the spinal cord as well as the forebrain. And he observed really interesting high levels of expression in the ventral tegmental area of a large number of these axon guidance genes in the adult nervous system. So we hypothesized that these genes are likely playing an important role in regulating the connectivity of the different inputs to the dopamine system. So what we wanted to develop is a method to do high throughput screening of these different genes to characterize which ones are functionally important. So in order to do that, before we get into the high throughput method that we're working on now, we first used just a conventional approach. So we wanted to develop a pipeline that we can use for later high throughput screening. So we decided to target Robo2. So roundabout 2 is an axon guidance receptor, which is critically important for the early establishment of the central nervous system projections by controlling the trajectory of axons as they pathfind on their way to their targets. So it turns out Robo2 is highly expressed in the ventral tegmental area in the adult mouse, consistent with the in situ data that was provided by the Allen Institute. So our first approach was just to knock out Robo2 and then run it through what we're creating as our pipeline for screening genes. So we got a Robo2 conditional mouse from Weining Liu and injected AAV-Cre into the ventral tegmental area to knock it out. We then looked at synaptic connectivity to see if there were changes in the synaptic connectivity of the ventral tegmental area after we inactivated this gene. So we looked at spontaneous inhibitory postsynaptic currents, spontaneous excitatory postsynaptic currents onto dopamine cells and determined whether or not there was a change. So when we knocked it out, we saw a dramatic reduction in the frequency of spontaneous inhibitory postsynaptic currents onto the dopamine cells. So Robo2 appears to be playing a role in maintaining the inhibitory synaptic connectivity of the dopamine system. What was interesting is it's actually controlling the connectivity of the inhibitory system, what would be predicted through a presynaptic mechanism, but what turns out to be a postsynaptic mechanism. So when we look ultrastructurally in animals that have intact Robo2 or inactivated Robo2, they are largely normal. The kinetics of their postsynaptic inhibitory responses are largely normal. The number of inhibitory GABAergic vesicles labeled are normal, but what we observe is a dramatic reduction in postsynaptic receptor density. So it appears as though all of the inhibitory synaptic connectivity remains intact, but the postsynaptic cell is no longer detecting the presynaptic events. Okay, so the next thing we do in the pipeline is if we've identified changes in synaptic connectivity, we look at changes in activity patterns in the awake behaving animal using standard electrophysiological techniques, in this case demonstrating that there is an enhancement not in the overall firing activity of the dopamine cells, but rather an elongation of burst firing events and a reduction in long loop inhibitory feedback, which we can elicit by injecting the animals with psychostimulant drugs. Furthermore, there is a dramatic increase in the phasic activation of the dopamine neurons whenever you inactivate this, and this can be measured by fast scan cyclic voltammetry. So we can show that there is a drastic reduction in inhibitory synaptic connectivity, results in vivo firing pattern changes, which results in vivo activity pattern release. And then the next question is, okay, is there a behavioral phenotype associated with this mutation? So what we observed here was sort of equally remarkable. The animals are hyperactivity, which is not that surprising given that we've removed GABAergic tone. They also appear to have learning deficits, which is not all that surprising since we've manipulated the dopamine system. They're hyper-dopaminergic and hyperactive. What was remarkable is the response of these animals to psychostimulants. So when you inject these animals with cocaine, amphetamine, or a dopamine selective reuptake inhibitor, they have an inverted locomotor response. So a normal animal, which is shown in black, increases its locomotor activity and responds to a psychostimulant. These animals are hyperactive and stop moving when you give them a psychostimulant. So for those of you who may know, this is remarkably similar to what happens in attention deficit hyperactivity disorder, in which you have hyperactivity, learning deficits, and inverted psychostimulant responses. So what we have recapitulated through a manipulation of this specific axon guidance gene is a manifestation of ADHD-like phenotypes. Okay. So we then went on to show that it's actually surprisingly not required in dopamine neurons alone, so you can inactivate the gene specifically in dopamine neurons either during development or in adulthood. It only is manifest whenever you activate the gene both in dopamine and non-dopamine neurons simultaneously. So Robo2 is controlling the inhibitory connectivity of the ventral tegmental area by acting on both local neurons that produce dopamine as well as non-dopamine producing. And we have evidence that this is likely to be largely mediated through this inhibitory synaptic connectivity, because when you selectively silence inhibitory transmission in the ventral tegmental area, you get the exact same phenotype. So you get hyperactivity and inverted psychostimulant responses. So the GABAergic control of the ventral tegmental area, which I demonstrated earlier, is arising from a large number of projection areas to the ventral tegmental area, plays a key role in maintaining the proper function of the dopamine system. When it's disrupted, you get attention deficit hyperactivity-like responses. Okay, so that's the pipeline. How do we achieve high throughput analysis to look at multiple different genes and really begin to map what is the genetic regulation of this functional circuit connectivity? To achieve this, we've begun using CRISPR-Cas9 mediated DNA mutagenesis through AAV-mediated viral delivery. So this was first demonstrated by the Zhang lab, where they targeted MECP2 through viral delivery of SPCas9, as well as the specific guide RNAs to target MECP2, and showed that they could knock it down. So what we've done is essentially the same thing. Now we're taking and generating sort of libraries of guide RNAs to different axon guidance genes and other synaptogenic genes. injecting them into the ventral tegmental area, and then running them through the pipeline to see if there are changes in synaptic connectivity. If there's changes in synaptic connectivity, we then move to the next stages of the characterization. So we can actually screen tens of genes at one time as opposed to one gene at a time, and then narrow down what are the key functional genes for the regulation of the connectivity of the midbrain dopamine system. And the first pass that we tried to test this was just targeting tyrosine hydroxylase, which is the rate-limiting enzyme in dopamine production. And by generating a mix of AAVs that contain guide RNAs to TH, we could reliably knock out TH on one side of the brain. So the strategy appears to be working, and we now have the first mice which are running through the pipeline with pools of guide RNAs to different synaptogenic molecules. So with that, I will take questions and acknowledge the individuals who did the work. So all of the mapping of the ventral tegmental area specific inputs was done by Amanda Zilla with help from Jesse Resnick and Samara Miller. The Robo2 project is guided by Brian Gore with help from Samara Miller as well as Amanda Zilla and several others. So thank you.
Yes, I have a question here.
Very interesting talk. I just wonder, when do you start to do viral transduction to knock out genes or to do CRISPR during, what stage? And do you know when do dopamine neurons establish these input-output connections during development?
So two great questions. So we don't know yet when the input-output connections of the dopamine neurons are firmly established. We know that the dopamine neurons manifest their molecular phenotype around E13.5. So that's when dopamine-specific markers begin to turn on and they begin projecting to their target areas. We assume that inputs from other areas are beginning to coalesce around that same time. So for us, when do we target the specific genes? So we're actually interested in figuring out what these genes do in the adult nervous system. So for Robo2, for example, we know that it controls growth cone motility. We had no idea that it controlled inhibitory synaptic connectivity. We figured it must be doing something because it's so highly expressed in the adult nervous system. So it was either vestigial or it's maintaining some function. Turns out it's maintaining some function that wasn't known previously. So we want to know, what are these genes doing in the adult nervous system? And for two reasons. One, because we don't know, and two, because a lot of these genes that we're interested in are highly implicated in mental illness. So of the genes that have been shown to be mutated in autism, bipolar, schizophrenia, the largest family is synaptogenic and axon guidance molecules. And we want to know is, these are probably early wiring deficits to some degree, but if they maintain adult function, then they may be tractable targets for therapy. Yes.
Oh, microphone.
So you said that the Robo2 is required not for these effects that you've seen. The knockdown is required not only in the dopamine neurons, but also in other neurons. And is this correct that
Yes.
these are the upstream neurons?
No,
that
so
are transmitting GABAergic signals to them, or other neurons?
So it's required in other neurons in the ventral tegmental area. So if you knock the genes out of the dopamine neurons, it doesn't do anything. If you knock the genes out of the dopamine neurons and non-dopamine neurons in the VTA, it emerges. So what we don't know yet is which specific inhibitory synapses are affected. Is it local inhibitory synapses that require Robo2, or is it the distal inhibitory projections that require Robo2? And we're trying to figure that out right now. Oh, thank you.
There's one in the back. One more question.
Yeah, I was just wondering if you could comment on, since they're spontaneously more active, with the psychostimulants, did they go into stereotypies and that's why they moved less, or was it really some sort of a calming effect?
Yeah. So that's a great question. And I have movies, but I didn't show them because I always get paranoid in short talks showing movies for things crashing and turning disastrous. But actually what happens is the animals sort of bounce around, and we actually have data looking at footprint analysis. And what the animals appear to do mostly is they kind of jump like rabbits most of the time. And then when you give them the psychostimulant, they just stop. So I can't say if it's actually calming, right, where they're more relaxed, or if it's some sort of catalepsy that has occurred. It does not appear to be catalepsy. Like if they decide to move, their movement appears to be relatively normal. And we've also looked, we thought, well, maybe if it's cataleptic or some other response, it might be aversive. So we looked at conditioned place preference to cocaine injection, and they find it rewarding to the same degree as the control animals. So we think it is actually a calming effect, but it may still be some sort of catalepsy that's emerging. We haven't figured that out yet.
Thank you. Thank you very much. And